Good day, everyone. So, for today's topic, it might take a while since this topic is a quite long process, okay? So, here we'll be talking about cellular respiration or how our body produces energy or what we call ATP through the cellular respiration. Okay, so when we say cellular respiration, it is how our cells make energy from the food that we eat, especially the glucose, or it is a type of sugar. So our cells, they cannot use the glucose directly. So this process or the cellular respiration will change it into a form that they can use. And that form is what we call the ATP or adenosine triphosphate. Okay, so this adenosine triphosphate is also known as the energy currency used by our cell, and think of it as the cell's money or cell's fuel that it powers anything the cells does, like moving muscles, transporting materials across the membranes, building proteins, and also repairing the damaged part. Okay, so the overall process of cellular respiration can be processed by this equation. Okay, so wait, by this equation. Okay, so this means that one molecule of glucose here, the C6H12O6, one molecule with um one molecule of glucose reacts with 6 molecules of oxygen in order to produce 6 molecules of carbon dioxide, 6 molecules of water, and an energy in the form of ATP. Okay, so this is the overall process, but again, we will discuss it thoroughly later on. Again, the equation for the cellular respiration is one molecule of glucose, and it is it is reacted with the or it reacts with the 6 molecules of oxygen in order to produce 6 molecules of carbon dioxide, 6 molecules of water, and an energy in the form of ATP. So in short, our cells take in food and oxygen, and in return, they will give us the energy to live and also to move. So here are the major stages or the 4 stages of the cellular respiration. Okay, so we have here the glycolysis, okay, the pyruvate oxidation, the Krebs cycle, or also known as the citric acid cycle, and lastly, we have the electron transport chain or ETC and oxidative phosphorylation. So in this diagram, we can see the summary, okay, or to summarize the whole process of cellular respiration. But again, for every process, we will discuss it thoroughly. So now let's begin with the first major stage of cellular respiration, which is the glycolysis. Okay, so when we say glycolysis, it is the process of breaking down one molecule of glucose. Okay, or when we say glucose, it is a 6 carbon sugar. And this glucose will be broken down into 2 molecules of pyruvate. And in this 2 pyruvate, each of them has 3 carbons. Okay, again, when we say glycolysis, it is the process of breaking down one molecule of glucose or this 6 carbon sugar, and it will be broken down into 2 molecules of pyruvate, and each of this each of this pyruvate has 3 carbons. Okay, so this glycolysis, it can occur in the cytosol of the cell. Okay, and this process or this glycolysis, it does not require any oxygen. So it means that it is anaerobic process. Okay, it is anaerobic. Okay, so here, what happens here is that, again, the one glucose molecule or the 6 carbons is broken down into 2 pyruvate molecules and 3 carbons each. So when this happens, a small amount of ATP or a small amount of energy is made. And aside from that, it will also produce NADH. So that NADH will be used later on to make even more ATP or energy. Okay? So this first stage, it is very important because it gives the cell quick energy when needed, and it also prepares the important materials like the pyruvate and the NADH for the next stages of the cellular respiration. Okay, so again, when we say glycolysis, it is the starting point of the energy production from the glucose. Okay, and also this glycolysis, it can be divided into two major parts or two major phases. Okay, so the energy requiring phase, okay, so this part, and the second phase is the energy releasing phase. Okay, so this one. Okay, so in short, the cell will first spend a little energy in order to earn a larger amount for the next steps or for the next stages. So let's focus first on the energy requiring phase, or also called as the energy investment phase. Okay, so here it is where the ATP is used to prepare glucose for breakdown. So the first step that we have here, or the glucose phosphorylation, in this first step, the enzyme hexokinase here will be added into a phosphate group from the ATP to glucose. Okay, so when this happens, when the enzyme hexokinase already added from the ATP to glucose, it will now form the glucose 6 phosphate, or the G6P. Okay, so this first step, it is very important, okay, because it will trap the glucose inside the cell, because the charged molecules like the G6P can't cross the cell membrane. So it makes the glucose more chemically active, okay? And this first step, it uses one ATP molecule. Okay, again, this step uses one ATP molecule. Okay, so again, that is the first step of the glycolysis. Next,
the enzyme phosphoglucose isomerase will now rearrange the glucose 6-phosphate into fructose 6-phosphate. Okay, so again, because of this enzyme, or the phosphoglucose isomerase, we can rearrange the glucose 6-phosphate into fructose 6-phosphate. So this simply changes the structure from one sugar to form another sugar. Okay? So, sorry if medyo nabago yung setup natin. Lumipat na lang ako rito para mas madaling magsulat. Okay? So, after the isomerization process, we will now proceed to the second phosphorylation. So, here, another ATP will donate a phosphate group, and they will donate a phosphate group through the enzyme which we call the phosphofructokinase, or what we call the PFK1. Okay? Again, another ATP will donate the phosphate group through the enzyme phosphofructokinase, and because of that, we will now produce what we call the fructose 1,6 biphosphate, or the F1 6BP. Okay, so again, the enzyme, through the enzyme phosphofructokinase, we can now produce the fructose 1,6 biphosphate. So this molecule now has two phosphate groups, one on each end. So because of that, it makes it very unstable. Okay, because of this, it will also be ready to split in half. Okay, so this is step. Okay, and this is step. It will use another ATP, bringing the total ATP used so far for 2. Okay, so in the second phosphorylation, we will use 2 ATP. Okay, so here, this is the main regulatory step of the glycolysis. So it will control how fast or how slow the pathway will run, and it depends on the cell's energy needs. Okay, again, in the second phosphorylation, or in this part, what we're going to do is that it is the main regulatory step of the glycolysis. Okay? So, it is like a checkpoint or control switch for glycolysis. So, now that the molecule fructose 1,6-biphosphate has become unstable, it will now be ready to split in half, okay? So, in order to do that, the enzyme aldolase does the splitting. So, here, as you can see, we have 6-carbon sugar. So, in fructose 1,6-biphosphate, it is a 6-carbon sugar, and now it will be divided into 2, okay? It will be divided into 2, and each of them has 3 carbon molecules. So again, this is the glycerol dihyde 3-phosphate and the dihydroxyacetone phosphate. And again, each of them has 3 carbon molecules, okay? But here's the catch, guys, okay? So, as you can see here, we have DHAP and G3P, right? But after that, the enzyme, wait, I will just write it. The enzyme triose phosphate isomerase. So, this enzyme will convert, or it will quickly converts the DHAP to another G3P. So here we will now have 2 G3P, okay? And both of the G3P molecules will continue through the next steps of the glycolysis, okay? So again, in this step, it is like cutting one big sugar into half to make 2 smaller pieces, or the DHAP and the G3P, and then afterwards, because of the triose phosphate isomerase, this DHAP will be converted to G3P as well. Okay, so that is the steps in the energy requiring phase. So next, we'll be focusing on the energy releasing phase, or what we call the energy payoff phase. So here, it is where the ATP and the NADH will be produced, okay? So here, now that we've spent energy to prepare the glucose for breakdown, it is time, or it is the time for the payoff phase, okay? It is where the cell will start to produce the ATP and the NADH. So each of the 2 G3P molecules, so I will just wait. So the DHAP again will now be G3P. Okay? So again, each of the 2 G3P molecules will undergo next steps, meaning the reactions occur twice per glucose molecule. Okay? So here in this step, or in the oxidation of G3P and formation of NADH, in this step, each of this G3P molecule will be oxidized, okay? It will be oxidized by the enzyme glyceraldehyde 3, or G3P dehydrogenase, okay? Again, each of this G3P will be oxidized by this enzyme, or the GAPDH. So, as G3P will be oxidized, okay, if the G3P will be oxidized here, they will accept, or the NAD here will accept electrons, and the not only the electrons, and also hydrogen ions, okay? So again, as the G3P will be oxidized here, the NAD will accept electrons and hydrogen ions in order to form the NADH. Okay? At the same time, when that happens, the inorganic phosphate is added into G3P. Okay? So again, when this is happening, as the G3P is oxidized and the NAD will accept electrons and hydrogen ions in order to form the NADH, the inorganic phosphate will be added as well to G3P in order to form this 1,3-biphosphoglycerate. Okay? So, this 1,3-biphosphate glycerate, it is a high energy intermediate. Okay? So, again, in this process, parang dalawa yung nangyayari, okay? So, the first one is that again, habang na-oxidize si G3P, yung NAD+, mag-a-accept siya ng electron and hydrogen ions para makapag-form ng NADH. And at the same time, yung inorganic phosphate is ma-add din siya sa G3P, and because of that, it will now form the 1,3-biphosphoglycerate. Okay? So, here, since we have two G3P molecules, we produce 2 NADH. Okay? Here in this step, again, since we have 2 G3P, we will now produce 2 NADH. Okay? So now, the cell will start to get the energy back. So again, in 1,3-biphosphoglycerate, here, we have 2 G3P, right? 2 G3P. So, each of the molecule here, or each G3P, will give off its phosphate group to ADP. So again, each of this will give its phosphate group to ADP. Okay, ADP. And if that happens, or when the ADP gains that phosphate, it will now become the ATP. Okay, so from ADP, and if it has the phosphate, it will now become the ATP. Or again, it is the main energy molecule of the cell. So, in order for this to happen, we use the enzyme, what we call the the phosphoglycerate kinase. Okay? So again, in order to do this, we use the enzyme phosphoglycerate kinase. Okay? So in this process, it is the first time that ATP is actually made during the glycolysis. So this process of making ATP is this process of making ATP by directly transferring a phosphate here is called the substrate level phosphorylation. Again, in this process here, it is called the substrate, I will just write, substrate level phosphorylation. Or it means making ATP from a molecule directly. Okay? So, again, since here we have two G3P molecules, we have two G3P molecules, it means that in this step, we make 2 ATP in total. Okay? So, 2 ATP in total, one from each of the G3P. Okay? So, after making the ATP here, the molecule we have now is called the 3-phosphoglycerate, or the 3PG. So, in this step, the cell rearranges it for a bit in order to prepare for making more energy. Okay, so the rearrangement here is very important to prepare more energy. Okay, so in this process, the 3PG, or the 3-phosphoglycerate, is will be rearranged into 2-phosphoglycerate, and it is rearranged by the enzyme phosphoglycerate mutase. Okay, again, this 3-phosphoglycerate is rearranged because of the enzyme phosphoglycerate mutase, and after that, the enolase here will remove a water molecule from the 2-phosphoglycerate. Okay, again, the enzyme enolase will remove a water molecule from this 2-phosphoglycerate, and again, it is a dehydration process because we remove a water molecule. Okay, so after that, after the enolase remove the water molecule from the 2-phosphoglycerate, we can now create, or we can now have the phosphoenol pyruvate, or the PEP. So this PEP, it is a very high energy molecule, and it is ready to make the ATP in the next step. Okay, so again, in this process, the phosphoglycerate mutase will move the phosphate, and the enolase will also remove the water in order to make the PEP. Okay? So, next is the final step of the glycolysis. So here, the pyruvate kinase will remove the phosphate group from the PEP. Okay? So, since the pyruvate kinase will remove the phosphate group here, the phosphate group will now transfer to ADP. And if the phosphate group from here will be transferred to ADP, we can now produce ATP again. Okay? So again, when the PEP gives away its phosphate, it becomes the ATP and also what we call the pyruvate. Pyruvate, okay? So when we say pyruvate, it is a 3-carbon molecule, okay? So since this happens, again, the phosphate is donated to ADP and it forms the ATP. It is also called as the substrate level phosphorylation again. Okay, so again, when we say substrate level phosphorylation, it means that the ATP is made directly from a molecule, not from the electron transport chain. Okay, so here, since we have two PEP molecules, it makes 2 ATP. Okay, so again, here we have 2 ATP and 2 pyruvate molecules. And these pyruvate molecules will now move into the mitochondria for the next stage, or what we call the pyruvate oxidation. So after glycolysis, the 2 pyruvate molecules will now move from the cytoplasm into the mitochondria. Okay? Again, the 2 pyruvate molecules from the cytoplasm will now move into the mitochondria. So there, each of the pyruvate will be converted into acetyl-CoA. Okay? And this acetyl-CoA will release carbon dioxide later on and forming NADH in the process. So this step, or this pyruvate oxidation, it serves as the bridge, okay? It is like a bridge between the glycolysis and the Krebs cycle, okay? So again, this step happens after the glycolysis and it takes place inside the mitochondrial matrix, okay? So specifically, mitochondrial matrix, or it is the innermost compartment of the mitochondria. So this pyruvate oxidation, it is considered as aerobic, okay? Aerobic because it needs, or it indirectly requires oxygen. So even though oxygen isn't used directly in this step, the next stage, or the electron transport chains, depends on the oxygen. So it allows this process to continue, okay? So again, the main purpose of the pyruvate oxidation is to act as the bridge between the glycolysis and the Krebs cycle, and also it prepares the pyruvate for breaking down by converting it to acetyl-CoA. Okay? So now let's proceed to the step-by-step process of the pyruvate oxidation. Okay? So here, after glycolysis, the 2 pyruvate molecules, each with 3 carbons, are formed in the cytoplasm. So these pyruvates must be transported in the mitochondrial matrix. So in order to be transported, a carrier protein or a transport protein in the mitochondrial membrane will help the pyruvate to move from the cytosol into the matrix. Okay, again, a carrier protein or a transport protein will help to move the pyruvate from the cytosol into the matrix. Okay, so once inside, the pyruvate is now ready for the next chemical changes. Okay, so inside the mitochondrial matrix, the enzyme pyruvate dehydrogenase will now remove 1 carbon atom from the pyruvate. Okay, so again, the enzyme pyruvate dehydrogenase will remove 1 carbon atom from the pyruvate. So this carbon is now released as carbon dioxide, or it is the first molecule of carbon dioxide that we exhale during respiration. Okay? So again, when we say decarboxylation, it is the removal of the carbon dioxide in the pyruvate, and it is removed by the enzyme pyruvate dehydrogenase. So, after this step, the pyruvate, which is originally had 3 carbons, is now reduced to a 2-carbon fragment. Because again, the pyruvate dehydrogenase removed the 1 carbon from the pyruvate. So, now that the pyruvate has 2-carbon fragment, okay, this 2-carbon fragment will be oxidized, okay? So, when we say oxidize, it loses electrons. So, during this process, the NAD plus will pick up the released electrons and also the hydrogen ions in order to form the NADH and also the hydrogen plus, okay, or the H plus. So, here, the NADH molecule will carry the high energy electrons into the electron transport chain. Okay, so if that happens, it will eventually help to generate the ATP. So this oxidation process not only prepares the pyruvate for the Krebs cycle, but it also captures the energy for the later use. Okay, so for the final step of the pyruvate oxidation, what will happen is that the oxidized 2-carbon fragment will now be attached in the coenzyme A. Okay, and this coenzyme A will now form the acetyl-CoA. Okay, so here the acetyl-CoA is now the key molecule. Okay, so this acetyl-CoA again, it is the key molecule that will enter the Krebs cycle. Okay, so it is where the majority of the ATP producing reactions will take place. Okay, so here again, this is why the pyruvate oxidation is called the link reactions, because it connects the products of glycolysis to the energy harvesting steps of the Krebs cycle. Okay, so again, in the final stage or in the last stage of the pyruvate oxidation, what will happen is that the 2-carbon fragment will be attached in the coenzyme A and it will now form the acetyl-CoA later on. Okay, so again, this molecule or the acetyl-CoA is the key molecule that will enter the Krebs cycle. So now that the pyruvate oxidation has produced the acetyl-CoA, we will now move on to the third stage of cellular respiration, which is the Krebs cycle, or also known as the citric acid cycle, or also the TCA cycle. Okay, so this is the stage wherein the energy stored in the acetyl-CoA is completely extracted and will be transferred to energy carriers like the NADH and also the FADH2. Okay, so here, the entire Krebs cycle also occurs in the mitochondrial matrix, or again, it is the innermost space of the mitochondria. And for this process, it is aerobic. Okay, again, it requires or it indirectly requires oxygen. So again, even though the oxygen isn't used in this step, the electron transport chain that follows will need oxygen to keep the cycle running smoothly. Okay, so again, this process is anaerobic or aerobic rather. Okay, it is aerobic. Okay, so here the main purpose of the Krebs cycle is to completely oxidize the acetyl-CoA into carbon dioxide. It also generate a high energy electron carriers like the NADH and the FADH2, and this electron or high energy electron carriers will drive the ATP production in the electron transport chain later on, and it also produces a small amount of ATP directly during this cycle. And lastly, it provides important intermediates that can be used by the cell to make the amino acids, fatty acids, and other biomolecules. So for each turn, okay, so for each turn of the cycle, it handles one acetyl-CoA molecule. But since one glucose molecule produces two acetyl-CoA, or from the one glucose molecule a while ago, we produces two G3P, right? So here, we produces 2 G3P. So, from there, we also have here 2 acetyl-CoA. And because of that, we will also have the cycle will also turns twice per glucose. Okay? So aside from that, we also have here inputs and outputs of the Krebs cycle. So for the inputs, we have the 2-acetyl-CoA, 6-NAD+, 2-FAD, and 2-ATP. And of course, we also have the outputs of the Krebs cycle, which are the 4-carbon dioxide, 6-NADH, 2-FADH2, and of course 2-ATP. Okay? So now we'll discuss the long process of the Krebs cycle. Okay? So let's discuss it step by step and thoroughly. Okay? So in the first step, the acetyl-CoA or the 2-carbon molecule that is formed a while ago during the pyruvate oxidation will enter the mitochondrial matrix. So this acetyl group or acetyl-CoA will now join the cycle, and they can join the cycle by combining with the 4-carbon molecule, which is the oxaloacetate. So again, the acetyl-CoA will join the cycle by combining or by joining with the oxaloacetate. So this step introduces the new carbon atoms from the glucose into the cycle. So later on, it will be released as carbon dioxide. Okay? So again, in the first step, the acetyl-CoA will join the cycle by combining with the oxaloacetate. So for the next step is the formation of citrate. Okay? So the enzyme citrate synthase here will catalyze the reaction between the acetyl-CoA and the oxaloacetate. And if this reaction happens, it will now form the citrate. Okay? So again, it happens because of the citrate synthase that catalyzes the reaction of these two. And if that happens, it will now form into citrate. Okay? So, because of this reaction, it will also releases the coenzyme A. So, this coenzyme A, it will also be reused later on. Okay? So, this is also why this cycle is known as citric acid cycle, because the citrate or the citric acid is the first stable product that is formed. Okay? So again, in the second step, because of this citrate synthase, we form the citrate. So for the third step, the citrate is rearranged to isocitrate. So in this step, the citrate is rearranged into isocitrate because of the enzyme aconitase. Okay? So here, what happens is that the hydroxyl group within the molecule will be moved to a new position. And because of that, because of the new position of the hydroxyl group, it makes the compound easier to oxidize in the next steps. Okay? So, this rearrangement may seem minor for this diagram, but it is very essential for the proper flow of the cycle. So again, in this third step, what happened here is that we experience rearranging of the citrate into isocitrate with the help of the enzyme aconitase. So the next step is the first oxidative decarboxylation. Okay? So in this step, the isocitrate, okay, will be oxidized, okay? And it loses one carbon atom as carbon dioxide. Okay? So again, in this step, the isocitrate will lose the carbon dioxide. And the enzyme isocitrase dehydrogenase will carry out this reaction. Okay? Again, because of the isocitrate dehydrogenase, we will remove or we can lose a carbon dioxide. So, as the electrons are removed, they are transferred to this NAD+. And if that happens, if the electrons are transferred in the NAD+, it will now form as NADH. Okay? So, this NADH will later be delivered as electrons in the electron transport chain. And here, in this step, the end of this step is that the molecule isocitrate will become the alpha ketoglutarate. okay? And here, again, in this step, we've captured our first NADH of the cycle. Okay? So again, it is the first oxidative decarboxylation. So next is we will experience or we will have the second oxidative decarboxylation. So here, the alpha ketoglutarate, okay, the alpha ketoglutarate will now be converted into succinyl coenzyme A. Okay? And it is converted by the alpha ketoglutarate dehydrogenase. Okay? So here, another carbon is released, okay, or another carbon dioxide is released. And of course, more electrons are transferred to the NAD+. And if that happens, if the electrons are transferred into NAD+, it will be converted to another NADH. So again, here we have another NADH, and for the remaining carbon molecules that is attached temporarily to coenzyme A, it will also form the succinyl-CoA. Okay? So again, at this point, both carbons from the original acetyl-CoA has been released as carbon dioxide. I mentioned it a while ago. Okay? So it means that the carbon backbone of glucose is now fully broken down. Okay? And it happens here in these stages, in the oxidative, first and second oxidative decarboxylation. So for the next step, or the substrate level phosphorylation, this is step, the molecule succinyl-CoA is changed into a succinate. Okay, so when this happens, the energy released from breaking the bond of the succinyl-CoA, okay, the energy or that energy is immediately used in order to make 1 ATP, or sometimes it's GTP. Okay? So, because of that, since the energy is immediately used to make 1 ATP, it is called as the substrate level phosphorylation. So, again, it means the ATP is made directly, not through the electron transport chain. Okay? So, the enzyme that helps in this process is what we call the succinyl-CoA synthesase. Okay, so again, because of this enzyme, it helps to change the succinyl coenzyme A to succinate. Okay, so in short, the formation of from the succinyl-CoA from the succinate has made a 1 ATP directly. But since each glucose makes 2 acetyl-CoA and the cycle happens twice, I've mentioned it a while ago, it gives 2 ATP in total from this step. Okay? So again, from this step, we produce 2 ATP again. Okay? So for the 7th step of the Krebs cycle, we have here the formation of fumarate and FADH2. So in this step, the molecule succinate here will be changed into fumarate, and it can be changed by the enzyme called the succinate dehydrogenase or the succinic dehydrogenase. So this enzyme, it is very special because it can work in 2 places, or it can be seen in both Krebs cycle and electron transport chain. So it is also why because of that, it is called the complex 2. Okay, it is called the complex 2. So here, as the succinate is changed into fumarate, the FAD or the carrier molecule will pick up 2 electrons and 2 hydrogen ions. And if the FAD accepts that 2 electrons and 2 hydrogen ions, we will now have the FADH2. Okay? So that FADH2 will later on transfer its electrons to the electron transport chain, and this FADH2, it helps to make the ATP, but compared to NADH, it produces little less energy. Okay, so again, this FADH2, it helps to make ATP, but it produces a little less energy compared to the NADH. So for the next step, so in this step, the fumarate, okay, it will turns into malate. And here, the molecule of water is added, okay? A molecule of water is added to fumarate with the help of the enzyme fumarase, okay? Again, the water is added with the help of enzyme fumarase, and because of that, we can have the malate. Okay, so here the water adds hydroxyl group to the molecule, and it also prepares it for the final oxidation step of the Krebs cycle. Again, for this step, the fumarate will turns into malate because of the addition of water molecule, and the addition of water molecule is added, or it happens because of the enzyme fumarase. And lastly, for the last step of the Krebs cycle, the malate here, okay, the malate will be oxidized back. Okay, it will be oxidized back into the oxaloacetate, and it is by the enzyme malate dehydrogenase. Okay, so in this step, the NAD plus will accept the released electrons and also will be formed as NADH. So again, the regenerated oxaloacetate is now ready to combine with another acetyl-CoA, and it will start the cycle again. Okay, so this regeneration of oxaloacetate will keep the cycle continuous, okay, as long as there's acetyl-CoA and oxygen available. Okay, so again, in this last step, the malate will be oxidized back into oxaloacetate by the enzyme malate dehydrogenase. So, to summarize, the energy yield of one complete turn, okay, just the one complete turn of the Krebs cycle, okay, so I'll just write it for you guys. So, we produced 3 NADH, 1 FADH2, 1 ATP, and 2 carbon dioxide. Again, for one complete turn only of the Krebs cycle and per acetyl-CoA. But since one glucose produces 2 acetyl-CoA, we will double these numbers for a total of 6 NADH, 2 FADH2, 2 ATP, and 4 carbon dioxide per glucose molecule. Okay? So finally, we will now discuss the final stage or the last step or last stage of the cellular respiration, which is the oxidative phosphorylation. So this is the stage wherein most of the ATP, about 90% of the total energy yield, is produced. Okay, so again, in this stage, it is where the most of the ATP is produced. And it occurs in the inner membrane of the mitochondria, specifically along the folds, which is called the cristae. Again, it occurs in the inner membrane of the mitochondria, or the cristae specifically. So, this oxidative phosphorylation, it has two connected process, okay? It has two connected process, and we have here the electron transport chain, or it is where the electrons are passed along a series of proteins and release energy. And we also have here chemiosmosis, okay? So here in the chemiosmosis, it is where the energy will form, or it is where the energy from those electrons is used in order to make the ATP. Okay, so again, it is two closely connected processes, but these two, they depend. These processes, they both depend on oxygen, which acts as the final electron acceptor. And because of that, since both of them uses or depend on oxygen, it makes them aerobic process. Okay, again, this oxidative phosphorylation, it is aerobic, or it requires oxygen. So here, the main purpose of the oxidative phosphorylation is to convert the energy that is stored in NADH and in FADH2. Okay? So these electron carriers, okay, so this NADH and FADH2, they are the electron carriers that are produced from the glycolysis and the Krebs cycle a while ago. And this two electron carriers, they will be converted into a large amount of ATP. Okay? So again, the energy that is stored in the NADH and FADH2 will be converted into a large amount of ATP later on. Okay? So again, these carriers, they donate their high energy electrons into this electron transport chain. So later we will discuss it thoroughly. Okay? And also, as the electrons will move through the chain, the energy is released and the hydrogen ions, as you can see here, the H+, okay, the hydrogen ions are pumped across the membrane. And this energy difference, it drives the ATP production. Okay, so in fact, in this stage, as I've mentioned a while ago, it produces most of the ATP. And in this stage, it produces around 34 ATP molecules. And that around 34 ATP molecules are formed from a single molecule of glucose. Okay, so again, that is oxidative phosphorylation. So we'll focus first on the electron transport chain, or ETC. Okay, so the electron transport chain, it is a series of protein complexes wherein it is embedded in the inner mitochondrial membrane. Okay, and the role of the electron transport chain is to transfer the electrons from NADH and FADH2 to oxygen, and it is done by a step-by-step series of redox reaction. Okay, so here again, the role of the ETC is to transfer the electrons from the NADH and FADH to oxygen through a step-by-step series of redox reaction. And also, each transfer, it releases a small amount of energy. And that small amount of energy is used in order to pump the protons or the hydrogen ions across the membrane, and it builds an proton gradient, or also known as electrochemical gradient. So let's first discuss the step 1, or the electron donation to the chain. So in the first step, the reduced coenzyme NADH and FADH2 will donate their high energy electrons to the chain. Okay? So the NADH will donate electron into the complex 1, or in the NADH dehydrogenase, and as electrons will pass through this complex 1, okay, the energy is released and it is used to pump the hydrogen ions from the mitochondrial matrix into the intermembrane space. Okay, so again, in the first step, the reduced coenzyme NADH and FADH2 will donate their high energy electrons to the chains, and the NADH will donate electrons to the complex 1 here. Okay, and it donates electrons because of the enzyme NADH dehydrogenase. And as the electrons will pass through the complex 1, the energy is released and it is used to pump the hydrogen ions from the mitochondrial matrix into the intermembrane space. Okay? On the other hand, in the FADH2, it donates electrons to the complex 2. And we use the enzyme succinate dehydrogenase in order to do that. Okay? So, however, the complex 2 here, it does not pump protons. Okay? Or it does not pump hydrogen ions. Which is why the FADH2 yields less than ATP than NADH, as I've mentioned a while ago. Again, the FADH2 have less energy or less ATP than NADH because in complex 2 is that it doesn't pump protons or hydrogen ions as compared to this. Okay? So, next is the electron flow through the chain. So after the electrons are accepted by the complex 1 and 2, they are transferred to a mobile carrier, which is called the coenzyme Q or the ubiquinone. Okay, so the scientific name for coenzyme Q is ubiquinone. So from coenzyme Q, the electrons are passed to the complex 3 or the cytochrome BC1 complex. Okay? And then another carrier called the cytochrome C, which move them to the complex 4 or the cytochrome C oxidase. Okay? So again, from the C coenzyme Q, electrons are passed to the complex 3 and then to another carrier called the cytochrome C, and then this cytochrome C will also move them to the complex 4 or the cytochrome C oxidase. And at each of this step or at each step of this chain, the electrons will also lose energy. Okay? So this release energy is also used by these complexes to pump more hydrogen ions. Okay? So again, the release energy is used by these complexes to pump more hydrogen ions from the matrix into the intermembrane space. So, because of this continuous pumping of protons, it will create a strong connection and also a charge difference across the inner membrane. And again, it is known as the proton gradient. Okay? So again, that is the step 2. From coenzyme Q, it is passed down to the complex 3, and then another carrier, which is the cytochrome C, will also move them to the complex 4. Okay? So again, that is the electron flow through the chain. So for the final stage or final step of the electron transport chain, so at the end of the chain, electrons will reach the complex 4 or again the cytochrome C oxidase. So here, the electrons are finally transferred to oxygen, and again, the oxygen is the final electron acceptor. Okay? So here, the oxygen will combine with electrons and hydrogen ions in order to form water. And it is formed through the reaction, which is the wait, I will just write it. Oh, andito pala. From, again, the oxygen combines with electrons and hydrogen ions in order to form water through the reaction or through this reaction. Okay? Again, this is the reaction in order to form water. So, this reaction, it is very important because it removes the low energy electron and also to prevent the chain from backing up. So, without this oxygen, if we don't have the oxygen, the entire chain here would stop because the NADH and the FADH2 could no longer unload their electrons. And of course, the ATP production will be lessened. Okay? So, which is why oxygen is essential or very important for our lives. Okay? So, again, that is the final electron transfer to oxygen. And again, we have here the it is where the oxygen will combine with electrons and hydrogens, and in order to form water, we will use this reaction. So, to summarize the electron transport chain, or here pala, it transferred the high energy electrons from the NADH and FADH2 to oxygen, and then it uses the energy to pump the hydrogen ions across the inner membrane. And because of that, it creates electrochemical gradient or the proton gradient that stores the potential energy. And after that, the stored energy or this stored energy will now be used in the next step, which is the chemiosmosis to actually produce the ATP. So now we'll be focusing on the chemiosmosis. So when we say chemiosmosis, it is the process by which the energy is stored in the proton gradient is used in order to synthesize or in order to make ATP. Again, when we say chemiosmosis, it is the process by which the energy that is stored here in the proton gradient will be used in order to synthesize or to make the ATP. So again, it refers to the movement of protons or hydrogen ions across or the movement of protons across the mitochondrial membrane or mitochondrial inner membrane from the intermembrane space back into the matrix. And again, they can be or they can be back into the matrix with the special enzyme which is called the ATP synthase. Okay? So again, the movement of protons across the mitochondrial membrane from the intermembrane space back to the matrix is made or it happens because of the enzyme called ATP synthase or this one. Okay? That's the ATP synthase. So here, this is where the cell will finally produces most of its ATP in this chemiosmosis. So now let's discuss the step-by-step process of the chemiosmosis. So here, we will have first the proton gradient formation. So here, the electron transport chain will continuously pump hydrogen ions from the mitochondrial matrix into the intermembrane space. So as a result, the intermembrane space has a high concentration of hydrogen ions which are acidic. So here, the hydrogen ions here are acidic. And the matrix has a low concentration of hydrogen ions. And they are more basic. So because of that, since here we have acidic and high concentration of hydrogen ions, and in the matrix we have low concentration and more basic hydrogen ions, this creates two kinds of potential energy. Okay? So it creates chemical energy and electrical energy. Okay? So when we say chemical energy, it is formed due to the concentration difference, and the electrical energy is created because of the charge difference. And together, this will form what we call the proton motive force. Okay? Again, because of the difference, okay, and because of the chemical and the electrical energy, we will now form what we call the proton motive force, which helps to drive the ATP production, or it helps to have the ATP production. So now the next step is the proton flow through the ATP synthase. So here, the protons, they now want to move back into the matrix. So here, they want to move back there, okay, in order to equalize the concentration. However, the inner membrane is impermeable to ions, so they cannot pass through here in the matrix. So the only pathway for the hydrogen ions is through the ATP synthase, or for this one, okay? So parang ito yung bridge nila para makapasok uli. Okay? So again, this ATP synthase is embedded into the membrane. So as the protons will flow through the ATP synthase, this movement will spins part of the enzyme like a molecular turbine. Okay? So here, the mechanical energy from this spinning drives will now join the ADP and also the inorganic phosphate in order to form the ATP. So again, the mechanical energy from the spinning drives will now join the ADP and inorganic phosphate in order to form the ATP. Okay? So this is the core event, or this step is the most important event of the chemiosmosis, because this is the direct synthesis of ATP, with the use of the energy from the proton gradient. Okay, so again, the proton or the hydrogen ions flow here in the ATP synthase because they cannot move across the matrix. So this is the only way in order for them to move across. And here, as the protons will flow or the hydrogen ions will flow to the ATP synthase, this movement will spins part of the enzyme like a molecular turbine. And if this happens, it will now join the ADP and the inorganic phosphate in order to form the ATP. And again, it is the most important event in the chemiosmosis. So the next step is what we call the ATP formation. So this reaction, it can be summarized as the ADP plus the which is the inorganic phosphate. Okay, so again, the ADP plus inorganic phosphate is equals to ATP. So because this process is powered by the oxidation of NADH and FADH2 through the electron transport chain, it is called the oxidative phosphorylation. Okay, and again, it is called oxidative phosphorylation because it is powered by the oxidation of NADH and FADH2 through the electron transport chain. And again, each of the NADH molecule contributes enough energy in order to generate 3 ATP, while each FADH produces about 2 ATP. So again, I will just write it. So again, each NADH molecule will contribute or it generates 3 ATP, while the FADH2, it produces about 2 ATP. So together, this process will have about 34 ATP molecules from 1 molecule of glucose. Okay? So second to the last step is the return of protons and completion. So here, once the protons have passed through the ATP synthase, they will accumulate again in the matrix. Okay? So meanwhile, the electron transport chain, they will just continue to pump new protons into the intermembrane space as long as the electrons and oxygen are available. Again, in ETC, they will just pump new hydrogen ions or protons as long as they have the electrons and oxygen available. So this continuous cycling, they will just maintain the proton motive force and also ensures a constant supply of the ATP. And finally, the last step of the oxidative phosphorylation as well as the cellular respiration is the regeneration and continuation. So here, when the NADH and the FADH2 donate their electrons to the electron transport chain, they are converted back into NADH plus and FAD plus. So, this regenerated coenzymes will now return to glycolysis and Krebs cycle. Okay? So, again, the NAD plus and also the FAD plus will now return to glycolysis and the Krebs cycle in order to pick up more electrons. And, of course, it will allow the entire process of cellular respiration to continue without interruption. Okay? So, that is the whole process of the cellular respiration. So again, that is cellular respiration. So as you can see, we will go through a lot of processes just to produce energy. So you use your energy wisely, okay? So wag niyong sayangin sa mga taong hindi kayang mag-reciprocate ng energy nyo, because making energy is hard work, okay? So I really hope you understand a lot from our topic today.